Hello grade 8s and welcome to the answer series Natural Sciences videos based on our study guides. This video looks at a practical experiment that allows you to test for the presence of carbon dioxide. We're going to do this experiment in two ways. First, with an animal, which will be me, and then for enrichment, we'll also look at how to do this with plants. For the first part, you're going to need a test tube or a small glass jar, a straw, and clear lime water. Clear lime water is an indicator for carbon dioxide, just like iodine is an indicator for starch. The clear lime water will turn a milky white color if carbon dioxide is present. To prepare the clear lime water, simply mix 1 teaspoon of slaked lime, you can buy this at your local pharmacy, with about 500 milliliters of water. Stir this well and then set the mixture aside to settle a bit. The excess slaked lime will sink to the bottom, and after a while I like to use a filter, just plain carton roll, to just ensure that the lime water that I use is very clear, removing any excess slaked lime. Pour about 20 milliliters of lime water into a test tube or a little glass bottle, and then use a straw to slowly blow bubbles into the clear lime water. The bubbles contain carbon dioxide from the exhaled air in my lungs, and you can observe that over time the lime water is changing to a more milky color. So this milky color is a positive test for carbon dioxide. And from this experiment we can therefore conclude that living organisms, especially in this case animals like me, release carbon dioxide during respiration. But what about plants? So to test this hypothesis in plants, you can either use a stem of a healthy plant or germinating seeds. If you opt to use an actual plant stem with leaves, you have to make sure that you put your final apparatus setup in a dark cupboard. Otherwise, the green parts of your plant are going to photosynthesize when they're exposed to light. And that's going to be a problem because with photosynthesis, it uses the carbon dioxide and won't be releasing it, which is what we need in this investigation. So to bypass this problem, I will be performing this investigation using germinating seeds, and you can buy these at your local supermarket. Germinating seeds are nice to use because they don't have any green chlorophyll-containing parts, which means that I can leave my apparatus setup in a nice warm place with sunlight to speed up this test. Other materials you will need include a test tube or a small glass jar, a permeable bag of some kind, for example a little mesh bag, a muslin bag, or I've opted to use an empty tea bag. Clear lime water that you've prepared, something to seal your test tube with, for example a test tube stopper, masking tape, or even if you're using a mason jar, you can just use the lid. Add about 20 milliliters of clear lime water to your test tube or glass jar. Place some germinating seeds into a permeable bag. Lower the baggie down into the glass jar just far enough so that your seeds are not touching the clear lime water. Secure the bag to the test tube or your glass jar, sealing it in some way so that no air can enter or leave in the length of this experiment. Now leave this apparatus setup in a warm place for at least a day and observe any changes in the color of the lime water. So what you'll see is that the clear lime water has turned milky white in color over time, and again this is a positive test for carbon dioxide. So from this experiment we can conclude that living organisms, in this case plants, release carbon dioxide during respiration. Check out the video description below for practice questions from our study guides. If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from the Answer Series, your key to exam success.